Okay, students, today we are going to discuss plasma protein. Please keep silence. Today we are going to discuss plasma protein. Students, we already know that in our body, the major circulatory systems are blood. The major circulatory systems are blood. Okay? So, how blood is formed? Blood majorly contain 50 to 60% water, 50 to 60% plasma, and other than 50 to 60% cellular components. Which one cellular component? RBC, WBC, and other cellular components. Okay? And in this plasma, when we go for the process of centrifugation, you already know what is the meaning of centrifugations. That means some liquid particles, some liquid solutions we can rotate at revolution per minute. Then their cellular component becomes settled down at the bottom with their density, and the supernatant, which is upper part of this test tube, upper part of this centrifuge tube is known to be as plasma. Okay? So the question is that what is the basic difference between plasma and serum? Yes. Clotting factors. Clotting factor that is mainly contain fibrinogen. When we exclude the fibrinogen from the plasma, it is to be called serum. Okay. So that is blood. Blood contain plasma, which is 50 to 60% volume by volume. Okay, because you know in our blood the major quantity of water is present. Okay. Now cellular component. Cellular component, it contains RBC, WBC, platelets. Okay. And platelets. Okay. And in this plasma, when we go for investigations of this plasma, we go for whole study of this plasma, what we get? We get two components. The first one is inorganic components. And second one is organic components. Organic components. In the inorganic components, plasma contain sodium, potassium, calcium, magnesium, calcium, magnesium, chloride, phosphates. Okay? And in organic components, it's mainly contain protein, it's mainly contain proteins, hormones, vitamins, lipids, and some organic metabolites like glucose, amino acids, and urea. Like glucose, amino acids, and urea. Like glucose, amino acids, and urea. So all these are the components of plasma. Okay? So somebody is asking how to define a plasma. Then we say it is a transparent, pale yellow, semi-aqueous medium which comes from the blood when we go for centrifugation process. When we go for centrifugation process, okay, then we got the plasma. Okay. Student, plasma is very important to study it about this because the major protein content which is present in the blood is present in plasma. Okay? Major protein content. We studied 3 major protein content. That first is albumin, globulin, and third one is fibrinogen. Okay, albumin, globulin, and fibrinogen. But question is that how will we identify it? How will we identify all these proteins which is present in the plasma? For this process, for the identification of these proteins, we go for the techniques, and the technique is known as, this technique is like electrophoresis. Okay? How will we identify by these techniques? The techniques are basically electrophoresis, okay, chromatography, chromatography, just a minute. Electrophoresis, chromatography, and Coon's fractions. These all are the techniques through which we identify these proteins, but in this all techniques, electrophoresis is a major techniques. Okay? Electrophoresis is what? Electrophoresis means there is the movement of this component when we provide electric field. Okay? Electro means electric field, phoresis means movement. When we provide electric field, then there is a movement, and according to their density, they can make the separate bands. On the basis of their bands, how will you identify? There are separate bands are appear, and on the basis of their density of these bands, whether it is lighter, whether it is lesser in color, we identify that this is separate proteins. Now we see the pattern basically. Okay. And this is generally a pattern film, and we call this film as electropheretogram. What we called electropheretogram. This contains two positions, anode and cathode. Okay. The first one major. अरे इसमें ये देखो क्या फंसा हुआ, निकाल के लाओ, दोबारा लाओ, कुछ फंसा हुआ थोड़ा निकाल के लाओ. First we got such patterns. Okay, this pattern is a pattern of albumin. Albumin. This is alpha-1 globulin. Alpha-2 globulin. This is beta globulin. Gamma globulin. And lastly, we get some quantity of their fibrinogen proteins. So students, you see this is the electrophoretic gram pattern in which you will see there is the density of this pattern, the density of this marking is too large. When we get the result of this pattern, there is light band or dark band will appear. Light band or dark band. Light band or dark band. Light band means the concentration of this protein is less, then we get light band. If the concentration of this protein is high, then we get dark band. Okay. Now, from this pattern, what do you think? The albumin concentration is high. Okay? The albumin concentration is high. When we go for this, we go for densitometer to assume this, but majorly the amount is here. That means albumin, it is 3.5 to 5 gram per 100 ml. Protein is only a parameter which is count in gram per dl. Okay, rather than other parameters majorly count in milligram per deciliter. Okay, this is the major differences when we calculate for this. Albumin, it is a larger concentration is present, then it present in less amount. After then globulin, it is globulin, it is 2.5 to 3.5 gram per 100 ml. Then alpha 2 globulin, it is alpha 2 globulin, it is 0.3 to 0.5. Then alpha 2, we have some confused, beta 1, globulin. This is alpha 1, this is alpha 2, sorry, yes. This is alpha 1, okay. And you write here alpha 1 globulin, alpha 2 globulin, it is 0.4 to 0.3 gram per dl, okay. Now beta globulin, that is here we make it beta globulin, it is 0.5 to 1.1 gram per dl. Gamma globulin, (0.82) per 100 ml. Okay. And last one, fibrogen, it is 0.2 to 0.4 gram per 100 ml, gram per 100 ml. Now students, see the pattern and the concentration of different plasma proteins. Okay, what we done here, we take the plasma and we run in the electrophoresis machine in different gel electrophoresis, okay, in which we get this pattern, and from this pattern we assume that this is the normal pattern of normal person. Understand? What I tell you, albumin is in high concentration, globulin is in low concentration, and fibrogen is very low concentration. How will you identify? By the pattern of these bands, whether it is dark band or light band. Okay? Why it is important to study about this pattern? Because in several disease, in several disorders, these pattern become change. Okay? And this pattern is helpful for the diagnosis of different diseases. Okay? इसको साफ कर दे। यहाँ पे ये कंफ्यूज मत होना, यहाँ पे क्या है ना, ये अल्फा 1 अल्फा 2 है, यहाँ पे मैंने दोनों का ये प्रॉपर पूरा एक ग्लोबुलिन का वो लिख दिया है, ठीक है, ये अल्फा 1 एंड अल्फा 2। ओके, नाउ इसको साफ कर दे। With the help of different densitometer, we can check the density of this pattern basically. Okay, and these pattern are helpful for the diagnosis of different diseases. Diagnosis of different diseases. Which one? The first one is liver cirrhosis. Liver cirrhosis. Second one, nephrotic syndrome. Third one, multiple myeloma. Multiple myeloma. Okay. Fourth one, lymphatic leukemia. And lymphatic leukemia, and last one is in congestive heart failure. Congestive heart failure. Congestive heart failure. Now, student, what do you think? How will we identify the disease from this pattern? Anybody else? Yes.
Sir, most of the plasma proteins are either synthesized by the liver.
Yes.
So, if for in case if it is liver cirrhosis, so you can see that the amount of albumin in the plasma protein will be very decreased. But since the gamma globulins are produced by the plasma cells, their concentration will not be high for this disease.
Very good. Your answer is so correct, but it is correct. In some of the disease, it is majorly all are synthesized from the liver. Or what happened in this disease, particularly in the liver cirrhosis, the AG ratio become revert. AG ratio. Understand? During LFT, इसमें प्लाज्मा प्रोटीन होगा. दूसरा वाला, फर्स्ट, अरे फर्स्ट पीपीटी. हाँ, देखो. Basically, liver sources in liver function test, what we check? We assess the function of the liver, and how can we check it? By the synthesizing rate of the liver. How much synthesizes? Okay, it is mainly synthesize albumin and globulin, and we call this protein as AG ratio. But in the disease of liver cirrhosis, these pattern become altered. The synthesizing rate of albumin is very less rather than globulin. The pattern become invert. Okay. So with this pattern, we can identify the person having what? Person having liver cirrhosis and other kind of disorders. In nephrotic syndrome, basically there are some, I already tell you in the class if you are attended, that during proteinuria, the albumin is secreted out from the urine. So due to this, there is also loss of proteins through the urine. Okay. So these are the disease which we identify by the pattern. Now we take one by one a protein and we discuss in detail. Okay, we take one by one. You up to this you have to understand what is the meaning of plasma. What is the meaning of plasma? It is transparent, thick, pale yellow color substance which is obtained from the blood, and it majorly contains proteins. It majorly contains what? Clotting factors. Okay, but something I left out. That something is also present. Can you tell me which things I forgot? Which thing is forget which is present in the plasma? Please tell. Water or है? बोलो. मेटाबॉलिक वेस्ट बताया तो था. यूरिया. ओके. But this some other things are also present in the plasma which I forget here. You can tell me. Anybody else? But it is not present in major portion. It is present in trace amounts. It is present in trace amount. That is peptide hormone. Which one? Insulin and glucagon. Insulin and glucagon, because peptide hormones and some enzymes like LDH. LDH stands for lactate dehydrogenase. Okay. LDH, creatine phosphokinase, CPK. Okay. Creatine phosphokinase, CPK, and alkaline phosphatase, and glutamate, pyruvate, transaminase. I had already taught you about these transaminase, what they do. In the amino acid property, I will tell you, it is what? It is proceed transamination reactions. And what is happening in the transamination reaction? There is a transfer of amines group. Okay, so these are the enzymes: glutamate, pyruvate, transaminase. These are also present in the plasma, but in trace amount. Generally in the traceable amounts it is present. So now we take one by one all the proteins and we discuss in detail. Okay. First one is albumin. It is a globular proteins. Firstly, it is a globular proteins and it is majorly synthesized by the liver. Okay. It is to be a complete protein. Why I tell you complete protein? Because it contains all essential amino acids. All essential amino acids. That's why we say it is a complete protein and formed by a single polypeptide chains. This is generally the properties of these amino acids. Okay. And what is the function of this albumin? Most of the students know about this function. Huh? Oncotic pressures. Yes. Maintain body fluid distributions and blood volume. The major function of these proteins are maintain body fluid distributions and blood volume. Okay, the normal range you already know. The normal range of these The normal range of this albumin is 3.5 to 5 gram per dl. This is the normal range of albumin if a person having low level of albumin we call hypoalbuminemia. What we call hypoalbuminemia. Okay. So these are the first sign. Okay. And sometimes there is also person having high range of this albumin we called hyperalbuminemia, but it is not majorly happen. It is in very serious condition. These are generally hyperalbuminemia is majorly in the case of dehydration. Okay, majorly in the case of dehydrations. But hypoalbuminemia is majorly in different conditions like in liver cirrhosis, in liver cirrhosis, liver are not able to synthesize proper albumin, okay? And in the case of nephrotic syndrome, oblique acute kidney disease, we call as AKD. The major of the albumin is lost in the urine during proteinuria. Okay? Major in the cause of urine. It is not viral hepatitis is major also a cause. Okay? Hepatitis, viral hepatitis, liver cirrhosis, the first cause, second cause is nephrotic syndromes, and third major causes is chronic PEM. Chronic PEM means it is protein energy malnutrition. Protein energy malnutrition. Due to some of the socio-economic conditions, due to some of the problems, you will see some of the childs are deficient of calories. Some of the childs are deficient of calorie, deficient of proteins. That is, this child are we categorized in mainly two diseases. First one is Kwashiorkor. Second one is marasmus. Kwashiorkor and marasmus. These are the prime symptoms of these two of the disease. Okay, where intake of protein is very less. Intake of protein is very less, either by the mother or by child. So we discuss in nutrition topic all the details about this protein energy malnutrition. Okay, in the case of severe burns, trauma, where we lost majorly plasma. Okay. Which we lost majorly the plasma. Okay, what happens if there is less albumin here? What happened? Because it maintains the oncotic pressure of so what happens if there is less edema? There is edema occur in the body. Edema majorly in feet and hands. When you see the child of Kawashorkar Mersmus categorized child, you will see then there is first thing you will see there is edema in their feet, edema in their hands. Okay, so we categorize this. It is by due to less of albumin. Due to less of albumin, what happened there? There is a retention of fluid inside the tissues, and due to this retention, there is puffiness of these tissues, and we call this puffiness as edema. Okay, one of the important inflammation marker. Okay. Now, second one is plasma globulin. We discuss one by one. Plasma. Now, function of this protein is what? It is helpful. It is act as a transporter. You understand what is the meaning of transporter? It act as a transporter. You simply understand, if you want to come in the class, if you comes in the class with your friends, just you think what I tell you. If you comes in the class with your friend, then your friends is to be called a transporter for you. It is same like that. The several body metabolites needs transporter, such like unconjugated bilirubin, calcium, iron molecules. These required protein for their transportation in the blood. And which protein? Albumin. These are the major function of this albumin. Okay. The first function is it acts as a co-transporter for albumin, pre-albumin and globulin, maintain plasmatic pressure, defense and clotting and fibrosis. Okay, it is non-specific carrier for hormones, calcium, free fatty acid and some of the drugs like penicillin, phenytoin. Okay, and major of the drugs required albumin for their proper functioning, for their proper treatment. Okay. So why this hypoalbuminemia cause? I already tell you, it is by the improper functioning of the liver, whether it is condition liver cirrhosis in during malnutrition, in protein energy malnutrition, increased losses of albumin during catabolism infection. Increased catabolism infection means when a person having chronic pyrogens, chronic fever, high grade fever, then also there is a loss of albumin, excessive excretion by the kidney. It is nephrotic syndrome, and the symptoms is we call albuminuria. What we call? Albuminuria. When particular albuminuria is present in the urine, it is called albuminuria. And how will we confirm it? By which test? I tell you previously, by which test we confirm? Heat coagulation test. I already taught you in the class, heat coagulation test. Okay, excessive loss in bowel, severe ones. Edema due to low oncotic pressure, what happen? They can retain in the tissues and they causes edema. Okay. And if if there is a less albumin is present, they also loses their functions. They also loses their functions. They are not able to transport any metabolites and other things. Okay. There is no clinical condition. This is only a one physiological condition. I already tell you physiological and pathological, both the conditions are different. So in this, there is only dehydration which causes hyperalbuminemia. Okay. Now, alpha plasma globulin we discuss in next class. Okay. About plasma globulin we discuss in next class. So student, what we are understand up to this, I summarize it. That is what is plasma, how can we separate out all the proteins, by which techniques, electrophoresis, and through this pattern we can understand it is electrophoretic gram. Okay, sometimes pattern is appear like that and sometimes it is in this graph condition. This is the normal pattern of electrophoresis. Sometimes its graphs is appear, such graphs we get it, and these are the pattern. You will see there is light band and dark band. How will you differentiate it? Definitely by the concentration. If there is high concentration, there is dark band. If there is light concentration, it is light bands. Okay, so these all are different proteins. If we major separate out it is transferins, macroglobulin, lipoproteins, beta lipoproteins, immunoglobulins, because proteins having different other fragments also. Transferrin is what? It is a co-transport protein for the transportation of iron. Globulin, immunoglobulin is what? It is a antibodies. Okay. If we separate out all the proteins, we can get more fractions in this pattern. Okay. So one by one we discuss. In the next class we discuss about globulin and some other proteins in detail. Thank you.
Cytoskeletal, this term must be remembered. Prottekta topics here, each and every topics had some distinguished word, right? In case of RBC membrane proteins, these are the not the simple protein. This is the cytoskeletal proteins. Then
And under the membrane, compost structure.
it not only under the membrane. This is a wrong concept. It is maybe in the cytosolic, it may be in the transmembrane, that not be inside the cell membrane. Suppose this is a RBC. এটা একটা RBC বয় আছে. Fine. 3D structure. So this is a membrane. This is a membrane. Right? This is a membrane. Membrane is lipid bilayer. হ্যাঁ? কিভাবে হবে সেটা? ঠিক এরকম করে থাকবে। এই হ্যাঁ তাই তো এগুলো নিজেরা আঁকবি so lipid bilayers are there. Now some proteins are there that may be transmembrane, that may be intercytosolic. এবার বল আর কি বলবি? Very good. This is the importance of the mainly cytoskeletal proteins. এটার একটা এক্সাম্পল দিচ্ছি তাহলে ভালো করে মনে রাখতে পারবি। একটা ব্রাইড, নিউ ব্রাইড, সে একটা ড্রেস পরেছে। কেমন ড্রেস পরতো সেই দেখিস না সিনেমায়, ঘাগড়া মতো, হ্যাঁ জানিস তো কি না বলছে কেউ কেউ, ও জানে না। কে জানিস না? মনে কর কোনো একটা বিয়েতে তাই সে কোনো একটা ওয়েডিংয়ে বিয়ের আগে এই ঘাগড়া মতো পড়ছে, হ্যাঁ কে দেখিসনি? না দেখিসনি তার কিছু এটা ভালো লাগবে না এটা এক্সাম্পলটা। সো সামথিং ইজ ওয়ারিং টু দ্য কভার অফ হিজ লোয়ার পার্ট, হার লোয়ার পার্ট, রাইট? নিউ ব্রাইড। এবার সাপোজ সাম সর্টস অফ কি বলবো অ্যান্টিসোশ্যাল এলিমেন্টস, হ্যাঁ তাকে তাড়া করেছে। সে এবার বিভিন্ন গলি দিয়ে যাচ্ছে। আচ্ছা অনেকগুলো দেখি এখন তোর সাথে তোর সব ইয়ে বেরিয়ে গেছে, অ্যামাজন প্রাইম ফাইম হ্যান ত্যান, হ্যাঁ সিম্পল ড্রেসই ধর, একটা ঘাগড়ার মতো একটা জিনিস পড়েছে। ঘাগড়াটা এই জন্য বলছি, দিস ইজ এ আওয়ার আরবিসি, বেসিক্যালি ও হচ্ছে একটা আরবিসি। সাপোজ হি ইজ আরবিসি, সো হি ইজ গোয়িং টু ইন টানেলস অ্যান্ড এভরিথিং স্লো এরিয়া, ন্যারো এরিয়াস, দেন সাম সর্টস অফ ব্রিক্স আর দেয়ার, সাম কি বলবো ওয়ার অ্যান্ড টিয়ার লাগছে, ছিঁড়ে যাচ্ছে চলে যাচ্ছে। তাহলে কি হবে? আলটিমেটলি ওর জিনিসটা খারাপ হয়ে যাবে। সে কি করবে? সে তার ওয়েন টু অ্যাভয়েড দিস থিংস হি উইল চেঞ্জ ইজ শেপ। সে কি চাইবে? যে টু কভার আপ অল দ্য টিয়ার পার্ট, রাইট? Similarly, whenever RBC goes in the narrow capillaries, the coronary capillaries is very small, heart, kidney. So these are narrow capillaries are there, narrow channels are there, narrow blood vessels are there. There must be need some oxygen also. So must be RBC must be there. The RBC contains the hemoglobin, And hemoglobin contains heme and globin. In the heme there is a protoporphyrin and iron, and iron binds the oxygen. Simple. So if must be passed to the RBC, then he will go in the narrow. In the narrow part, if some tearing part is there, tear যদি ছিটতে থাকে, then this will transform into a globular part. তাহলে কি হয়ে যাবে? Shape of damage, shapeটা change হয়ে যাবে. What is the shape of RBC? Normal shape, biconcave. This is not sphere. The spherical shape is there. That means spherocytes will be there, and that may transform into the one disease is called hereditary spherocytosis or hereditary elliptocytosis. I am coming if time permits. Now these cytoskeletal proteins are role to এই তুই কোথায় যাচ্ছিস চলে এদিকে, এই তুই কোথায় যাচ্ছিস চলে এদিকে. They are pulling to avoid the tearing, and so the shape cannot be changed. Normally আমার তো সবারই, কিন্তু if anybody has some protein problems, genetic problem, gene problem for the ankyrin, spectin, 4.2, তাই তো 4.2 band 3, 4.1 protein, 4.2 protein, glycophorin A, glycophorin C, all proteins are very important. There are lots of protein during the erythropoiesis. Some functional, some are non-functional, but remember always 3 to 4, 5 proteins, that is important. One is pectin, another is actin. Actin also very important, it is intercytosolic. I am saying that this pectin is like that, right? কে কে পড়েছিস pectin structure? Pectin like that, এরকম থাকে. Dimer basically is a form of tetramer. এর এন্ডে এখানে হেড আবার এখানে হেড, and they are forming like that again head. So this is a tetramer form is the present, but in the tail form it is connected with a protein that is actin. This is called actin myosin, say actin. Okay, so these things are very much important. Now then you see that pectin is not connected to the membrane. এখনো কানেকশন আছে মেমব্রেনের সঙ্গে? কোনো কানেকশন নেই, আমি কানেকশন করিনি. So directly there is no connection with the cell membrane with the pectin, but it is very much important to do something to connect the membrane.